Hello and welcome to Global Biology Lesson. Today we are going to discuss the remaining questions for the topic biotechnology and genetic engineering. So let's start the question. The diagram shows the bacterial cell that is used to produce human insulin. We want to use this bacterial cell and we want to produce human insulin. So these are the objectives, right? A diagram is shown where they have shown a cut plasmid ring. This is actually a cut plasmid ring. Cut plasmid ring. They are asking that what is inserted in the gap Q. Okay? If I want to make human insulin, so it means that I will write the DNA from a human cell. This is our human cell with lots of chromosome in it. I will extract a segment of DNA from here that is responsible for the production of insulin. I will insert over here. So now let's see what are the options here. A gene from a healthy human, okay, the cell from the human pancreas cell. Okay, in this gap we cannot put the cell, right? DNA from another bacterium. A bacterium cannot produce insulin, so that's not the case. The molecule of insulin is actually a protein. So according to orbital arm technique, we are going to put the segment of DNA, the set of genes that are actually encoding for protein. We will pick those set of genes that are capable of producing protein. So this is actually the correct answer. Question number 12. Ligase is an enzyme which is used in genetic engineering. Hopefully, you have seen my first video of biotechnology and genetic engineering where I used the tape and scissor theory. Remember, if we want to cut the enzyme, so we are using the restriction enzyme. And if we want to paste a gene, sorry, if we want to cut a gene, we will use restriction enzyme. And if we want to paste a gene, we are going to use the ligase enzyme. The ligase is used to insert to join DNA into the DNA plasmid. Now, question number 13. Why are the bacterium useful in biotechnology and genetic engineering? Right. So, case study for the production of insulin, we have seen the bacteria are used for biotechnology. So that's not the case that bacteria do not have a vacuole, not having a mitochondria, not having a cell wall. These are not the criteria. The basic criteria is that bacteria share a genetic code with other organisms. Like there is some resemblance in the genetic makeup of bacteria and the other organisms. Just for that reason, we can use bacteria. There are multiple reasons as well. Our next question is based on the other reason. This is the reason for the bacteria to use in biotechnology. So bacteria are found inside the human body, not that the bacteria can become resistant to antibiotics. I will never pick any organism for that reason. Bacteria can make complex molecules. Like we were using bacteria and we were making insulin. Right? The bacterial cell was insulin is a complex protein. So bacteria can make a complex protein, that's the reason. And bacteria do not reproduce slowly, actually reproduce faster. So we have seen the correct answer here as well. And then our next question is based on the use of in baking. Why are we using yeast? Because yeast produces carbon dioxide that make the dough rise. Okay. Then in the next case, a crop plant has been genetically modified. For example, if we have a typical normal crop plant, and we are using a typical we have a typical crop plant, right? This crop plant has been modified and it has become resistant to herbicides. The herbicide do not affect on them. Okay. This is the possible disadvantage of introducing this new crop plant. If we introduce this crop plant in a whole field there are other non-modified. This is the modified plant and these are the non-modified plants. So what will happen? We know that the plant can do the process of cross-pollination. Right? Within this entire field, there might be some weeds also. Right? If during if during the process of cross-pollination, the herbicide resistance genes are acquired by the weed plant. So what do you think? What will happen to the weeds? Will they be killed by herbicide immediately, or will they not be killed? Like if a weed acquires a gene for herbicide resistance, so it will become resilient one. And if you are spraying any amount of herbicide on it, so your wheat plant will keep on growing faster and faster, and it will become uncontrollable. Okay? So this is the case that some weeds might become resistant to herbicide. Okay. Our next question is, which is the our next question is, this is the example of genetic engineering. Altering the gene DNA, the crop plant, they become resistant to herbicide. We are modifying the DNA to make any plant resistant to herbicide. This is a best example uh, of genetic engineering. Rest of them do not match with the criteria. Question number 8. What is inserted into the bacterium to become bacterium produced insulin? So, in the other previous cases of genetic engineering that we have discussed, we are actually inserting a length of DNA from humans, and we are picking that segment of DNA that can produce that encodes for insulin. Then our next question is, our next question is, genes are isolated from human DNA using dash enzyme. If we want to isolate a gene, so we are using which enzyme? Remember this is the rent state model. So it can be the restriction. It is actually the restriction enzyme which is to isolate a gene. So one can doubt for answer C or answer D. We need to verify the other options. The big fragment is cut with the same enzyme, forming dash. When we are, this is the human cell and this is the bacterial plug, right? Here using the restriction enzyme to make a cut in it. Here we are making a cut in it. We are making a cut in it. We design the cut in a way that it creates the sticky end. What is meant by sticky end? We are not getting into the detailed biochemistry. Sticky ends are the structure when we apply this enzymes, they tend to stick together. So formation of the sticky end is the reason. we cut the human DNA and plasmid in a in, uh, in the respective cells to make the DNA. The human DNA is inserted into the bacterial plasmid using an enzyme. So this enzyme is space the ligase forming a dash plasmid. Now the bacteria will be modified. It will not be purely made up of bacterial DNA. It will be bacterial DNA as well as some portion of human DNA. So this type of DNA, which is a combination of two different organisms, bacteria and or two different organisms, this is called recombinant DNA. This tend to be the correct answer. Okay. Our next and the last question for this topic is, which enzyme is used to make apple juice clear? We had a discussion. We make apple juice. So sometimes the pulp is present within the juice to make it unclear. The pulp contains many apple cells joined together. And how are they joined together? They are joined together with a substance pectin. It is actually a depositing material between the cells, like a cement. So if I want to make the juice clear, I will use an enzyme, pectinase. This pectinase will break down the pectin and release the bound cells together to release them into the free pregnancy. And this will result in making the juice more quantity and the juice will be clear also. So which enzyme we will be using? We will be using the bacterial enzyme. That's all from paper 1, and we are coming up with the next series of chapter questions. If you have any queries, I appreciate that you share your comments and the questions in the chat box and in the comments. Thank you for listening.